Hello Year 12, so today we're going to look at action potential. So we're going to start off by making sure you can remember what resting potential is and how it's generated. Then we're going to state what action potential is, describe the sequence of events. We're also going to look at threshold potential and the all or nothing response, and then we're going to get you to start to think about this last statement here and a couple of other things. So first activity I'd like you to do is answer these questions. You saw these last lesson, they were your thinking questions from last session. So can you answer these in 5 minutes? Pause the video. Okay, so let's see what you wrote for generating resting potential. So hopefully you got the idea that the plasma membrane is impermeable to sodium ions but more permeable to potassium ions. You could have had information about the gated channels in there, but you want the idea of differential permeability. You need the idea of the sodium-potassium pump with the unequal pumping of sodium ions and potassium ions, so 3 sodium out, 2 potassium in, active transport process. We've got some of the potassium ions diffusing out through the open potassium gated channels, and you could mention down their electrochemical gradient. Sodium can't diffuse back in, and what we end up with is a high concentration of sodium outside the axon and a high concentration of potassium inside the axon. And our resting potential is about 70 millivolts. Okay, so you could have had 60, you could have had 65, it depends on the organism. Suggest why the term resting potential is misleading. Well, resting just means not sending an action potential at that time, but it is still an active process because active transport is going on to maintain it using the sodium potassium pump. So respiration, aerobic respiration must be occurring to produce ATP, okay, which means we'd expect to find lots of mitochondria inside our axons because energy needs to be released for ATP to be produced for use in active transport. So there'll be lots of aerobic respiration going on, and remember, mitochondria are the site of aerobic respiration. So on a similar vein, what would happen if a metabolic poison like cyanide was used on the cells? So key thing, and we'll find out this out when we do aerobic respiration in detail, is cyanide prevents aerobic respiration, which would mean less ATP or no ATP being produced, which would mean active transport would stop or slow, which would mean you would lose the resting potential. You wouldn't be able to maintain it using the active transport proteins. Okay, and therefore if you don't have resting potential you can't get an action potential, so you can't send nerve impulses, and effectively if you're a complex organism like a human you would die. So let's have a look at what we mean by an action potential. So I showed you this graph last lesson, we had these regions, we're going to look at these. So the key thing here is we've got this change. We look at what's happening. Here's resting potential. We've got this change, this rapid change. We look at here, this is milliseconds, this is thousandths of a second. So within basically 2,000ths of a second, you have had this rapid change from -70 up to about +40 if you look across the scale, and then returning very quickly back, actually going slightly more negative than resting potential, and then returning to resting potential. Okay, so action potential is this rapid change in the potential difference across the membrane from this -70 from our resting potential, -70 to +40 millivolts and then back again. And because it is a potential difference, so it's +40 millivolts, but it's that change. Okay, and we talk about depolarization followed by repolarization, because remember we said our membrane when it's at resting potential is polarized at -70. So here's our resting potential. Something happens to cause the membrane to depolarize, so we lose the negative charge and it gets less and less negative. Now it actually goes past 0 millivolts and then heads toward positive charge on the inside compared to the outside, but it's still called depolarization even though it's polarizing towards the positive, because polarized is -70, so it's moving away from -70, depolarization, and then we reach this +40 and that's our action potential. Then we get repolarization, so the charge returns back towards resting potential the voltage goes back towards being negative on the inside of the axon, but actually it overshoots and becomes more negative, and that's called hyperpolarization. Okay, remember hyper means more, so more polarized than normal. And then eventually it returns to resting potential. Now what you might want to do now is make a couple of notes from the last two slides on this and annotate this up, and then we'll talk about what's going on. Okay, so I'm going to talk you through the stages now. So you might recognise this diagram slightly modified from last time. I've ignored all the negatively charged structures like chloride ions and proteins, things like that. We're just looking at the positive ions. So here is our membrane, axon membrane. Here's our tissue fluid surrounding the axon. Here's the axoplasm. Here's the plasma membrane or cell surface membrane of the axon. These are our sodium gated channels, these are our potassium gated channels, and here's our sodium potassium pump. So here we're starting off at resting potential. So we're starting off with a high concentration of sodium outside the axon and a higher concentration of potassium inside the axon. Okay, so the membrane's polarized, as I said before, at minus 70 millivolts. Okay, now let's look at what's going to happen next. So depolarization occurs. Now, a loss of polarization to something called threshold causes the sodium voltage gated channel to open. So you can see here, I'll just reclose it. Okay, sodium voltage gated channel to change shape and open. And that means some sodium ions will start to move in down their electrochemical gradient, which will depolarize the membrane further. So this thing of threshold is really important, and it was actually that line E on the previous graph. You have to reach this threshold of depolarization, and we'll talk about how that occurs in later lessons, and that will trigger these sodium voltage gated channels to change shape and open. Notice they didn't all do it at the same point, but when we reach threshold, some will start to open and some sodium ions moved in, which depolarizes the membrane further than it was. So here's the same image, just copied again. So that depolarization then causes more sodium voltage gated channels to open, so more sodium ions move down their electrochemical gradient into the axoplasm, and that leads the membrane to become depolarized and reach action potential plus 40 millivolts. So the fact that you had that initial depolarization causing some voltage gated channels to open, which caused more depolarization, which caused some more voltage gated channels to open, is actually a small example of positive feedback. Okay, we've got a little loop there where the change caused a bigger change. However, once we reach that action potential, that voltage of plus 40 millivolts actually causes the sodium voltage gated channels to snap shut, so no more sodium can move in, and causes potassium voltage gated channels to open, and potassium ions will start to diffuse out down their electrochemical gradient. Okay, and you can see here we had positive ions moving in, which made the inside more positive, and then we got positive ions moving out, which means that positive charge on the inside gets lost. Okay, so the membrane starts to repolarize, okay, and go back towards being negative again. Now, the key thing is that the voltage gated channels for potassium don't change their shape again until the voltage, the potential difference, has gone more negative than resting potential. So that means actually more potassium ions move out than sodium ions moved in effectively. So you end up with the membrane becoming more negative than resting potential, and that's your hyperpolarization. Once that occurs, the voltage gated channels close. Now you'll notice now that all our ions are the wrong way around. All our voltage gated channels are closed, and all our ions are the wrong way around. So we've got the potassium ions have moved out to the outside, the sodium ions, some of them have moved to the inside, so we haven't got our concentration gradients the right way and as steep as we had before. So now you have to return to resting potential, so you have to use the sodium potassium pump to restore the balance of ions across the membrane. So there's our unequal pumping of ions out across the membrane. Okay, some potassium ion channels are open again, so some potassium ions will move out as before to restore the resting potential. Okay, so those are your sequence of events. Now what I'd like you to do is, you've got a cut and stick activity which talks through the stages of action potential. You could do that, or you could write your own bullet points from the previous slides. But what you should do is do that, review it, close your book, and then try and write your own bullet points again. So a bit of very early retrieval practice, but practice writing it out without looking at your notes. See how much you can remember from that short term, but practice is important. Give yourself 10 minutes or so to do that. Okay, I'm going to talk to you now about a couple of features that are really important about the action potential. I've already talked to you about this idea of threshold, the threshold potential. So you should talk about threshold potential really, rather than just threshold, and you might want to make sure that's clear in your notes. So it's the depolarization required to trigger the voltage gated sodium channel to open. Okay, and that then leads to depolarization to action potential. If you have a depolarization that doesn't reach threshold potential, you don't get an action potential. And all action potentials are exactly the same size, or their amplitude, how the change in voltage is always exactly the same. Okay, that's really important. That's called all or nothing. So you either get an action potential in full because you reach threshold, or you don't. Okay, so we look at this graph here. It's very similar. There's our threshold potential. So here we've got a number of overlays here. So we had a little depolarization for some reason, but it didn't reach threshold, so we didn't get action potential. And in fact, what would happen here is the sodium potassium pump will just restore that that depolarization back to resting potential. Here we had a slightly different, bigger one, but again didn't reach threshold, so nothing happened. In the third example, third way I'm talking, it reached threshold, and then we get our action potential, which depolarization, repolarization. Now I'll talk to you about refractory period next lesson, and I'll talk to you about hyperpolarization next lesson. Okay, so but it's really important that you get this idea of all or nothing. Now that might lead to some questions in your mind that you might want to think about, because an action potential is always the same size. You can actually think of action potentials being digital, which is like digital signals are like ones and zeros, so on or off. So you either get it, a signal, or you don't. Okay, well, action potential is like that too. So your nervous system is a digital system, okay, in terms of communication. So make some notes about that now, and then come back for the last couple of questions to think about. So, some questions I want you to think about ready for next lesson. Because all action potentials are the same size, how can you distinguish between a strong or weak stimulus from a receptor, or a strong or weak response, so making your muscles contract gently or strongly? Okay, so how does that work, given that it's an action potential is you have an action potential or you don't? How do we get depolarization in the first place? That's also important. You know, we think about in the middle of an axon, how do we get depolarization at that point to make it reach threshold? Okay, how does that action potential move along an axon? It was started at one end, either at the receptor end if it's a sensory neuron, or from the relay neuron, or wherever it is. If it's a motor neuron, it would have started there. How did that start in the first place? How does it move along the axon? Okay, and what is hyperpolarization for? We had this idea that potassium ions more potassium ions move out and sodium ions move in. It goes, more importantly, it goes more negative than resting potential. Why is that important? So obviously you can have a think about that. You want to have a read ready for next lesson. Okay, thank you.